So it gives me great pleasure to introduce tonight's keynote speaker, Alan Rein. I think Alan is a very fitting speaker, as he is a true friend of the Cold Spring Harbor Retroviruses meeting. So he organized the meeting himself in 1995, and I'm not sure if he's missed a meeting since, but definitely not in the last 10 years. So he's sitting in an unusual seat tonight. He usually sits just about up there, and I think pretty much every meeting I've come to, he's sat there, except last night I saw you'd moved up a few rows and along. I was like, very happy to see that you're still flexible and open to change. So here is a picture of Alan that I think was taken, 2, maybe 3 years ago? And I think let this serve as a reminder, actually, to all you postdocs and students out there that you must sign up your name to the sheets with all the photos on that'll be up on the boards probably tomorrow. Just so that when you give your keynote talks in a few years' time, we've got some historical photographic evidence with which to embarrass you. So I don't think this was actually Alan's first time at Cold Spring Harbor, though. So I think he's going to mention it, but Alan first came here not for a retroviruses meeting, because I'm not sure they existed then, but in 1961. So I'll do the calculation for you. That's a very impressive 55 years ago. So I'm sure one or two things have probably changed since then. But I suspect that there's no one in the audience that beats that. So you can call out. If anybody in the audience came here before 1961, I'm looking mainly down in the front row. No? Right, well, Alan, you win the award. And that comes with a drink in the bar later. So I first met Alan the first time I came to Cold Spring Harbor, and I was a PhD student having a poster on FE1. And Alan came to my poster and introduced himself. And I recognized his name from some of the great papers that were published in the 1970s on FE1 that I used to refer back to. And I was very impressed that these important people would come and talk to me at my poster. So I hope some of you students had a similar experience or will have tonight. So FE1 was just one of the many things that Alan has worked on, but I'm not going to give half his talk, so I'm going to let Alan tell you about all the other things he's worked on over the years. So I will just give you a brief outline of his career and where he's been. So Alan did his PhD at UC Berkeley with Harry Rubin, and we're going to hear more about that tonight, I think. He then did postdoctoral research as an American Cancer Society Fellow with Sheldon Penman at MIT. And then since 1976, he's been at the NCI. So he has been head of the retroviral genetics section in the ABL Basic Research Program from 1984 to 1999. And then he joined the HIV drug resistance program as head of the retroviral assembly section. And in fact, that's where he is now, although you've changed your name, right? So it's the HIV dynamics and replication program. So you've moved. So Alan's been on the editorial boards of several journals and currently includes Virology and Journal of Virology. He won the NIH Directors Award in 2012. And in 2014, he was elected a Fellow of the American Academy of Microbiology. So we're delighted that tonight he's going to give us some perspectives on retroviruses. Over to you, Alan.
Okay, thank you very much, Kate. That was lovely. So, retroviruses, some perspectives. What on earth does that mean? What is he talking about? So, the talk got that title because maybe 6 weeks ago, Kate asked me for a title, you know. And at the time, I had no idea what I was going to talk about. So I made up the vaguest title I could. And then, as it turns out, this is what you're going to hear. Retroviruses, some perspectives. And so a lot of the talk is going to be some history of the field, some will be kind of up close and personal, and some will be broad brush strokes, a decade at a time. I'll then say a few words about retroviruses and about viruses in general and how they may fit into the world of living things. And finally, I'll say just a little bit of personal stuff at the end, giving credit where credit is due. So Kate's right. I first came to Cold Spring Harbor in 1961. That's me on the left. You might not recognize me, and that's Arthur Chavnik on the right. He was director of the lab at the time, and I spent the summer doing Drosophila genetics in his lab. That's us looking at some Drosophila. And in the course of the talk, you'll hear a little bit of commentary from this guy. I just want to say, at least for me, at least in my heart, there is no place in the world like Cold Spring Harbor. And coming to this meeting is really like a pilgrimage for me. And well, I'll mention Cold Spring Harbor a little as the talk goes along. Okay, so looks like a slide disappeared, but anyway, yeah, I'm going to tell you about my PhD mentor and the virology done in his lab. That's him, Harry Rubin. I studied with him at Berkeley, and I'm going to describe the virology that was done in his lab while I was there. How many of you have ever heard of Harry Rubin? Some, yes, very good. So around about 1970, I think, he left virology and moved on to do his own brand of cancer research. And so there are a lot of virologists who have never heard of him, but he made very important contributions in the period when I knew him. So I went into his lab in 1962, and so as it turned out, in 1962, the symposium here was on animal virology, and probably the preeminent animal virologist was Delbecco at Caltech, and then another eminent animal virologist was Harry Rubin. What that meant was Delbecco's students and Rubin's students got to come to the symposium and wait on tables. And that's me, and that's me observing from a safe distance during the symposium on animal virology. And I just got to tell you, it was just amazing. So, you know, there was this little contingent from Berkeley, Rubin's students, and we came to the meeting and met this little contingent of students from Caltech, Delbecco's students, and I just couldn't believe it. They were so different. And I think, you know, although you can't really tell from these pictures, in general, we from Berkeley basically tried to look as disreputable as possible. And I just expected, you know, the students from another distinguished university in the same field a few hundred miles away would look the same. But no, they were different. Let me tell you, they had tight pants, big wide belt, fancy buckle, wraparound shades. And I figure that the revolution was coming to Berkeley. That we were the vanguard, maybe, of the revolution. And I don't know if the revolution ever came to Caltech, but it sure hadn't come in 1962. Anyway, but we digress. I'm going to talk to you about Rous sarcoma virus. This is the beginning of the story I'm going to tell you. So in the early years of the 20th century, Peyton Rous made this very basic discovery. He took a chicken tumor, ground it up, made a cell-free filtrate from it, and injected it into another chicken, and the other chicken got a tumor, got a sarcoma. So that was Rous sarcoma virus. And again, now I'm going to talk to you about the virology that was done on Rous sarcoma virus in the early 60s, mid-60s, or actually beginning in the late 50s. So Ruben actually in the middle 50s, for a lot of the 50s, was a postdoc in Dolbecko's lab at Caltech. And it was known already from the 1930s that if you put Rous sarcoma virus onto the chorioallantoic membrane of chicken egg, it would induce these little microtumors, or pox they were called. So this is a section of uninfected chorioallantoic membrane, and this is one of these little tumors induced by Rous on chorioallantoic membrane. And having something to count, namely these little tumors, made it possible, in a very rather primitive way, but made it possible to do quantitative virology. And so this was published in 1955. This was a really striking, important result. So Ruben did a microdrop experiment. That is, he put Rous sarcoma cells into tiny little microdrops and put them on a microscope slide submerged in oil. And he could observe the cells in the microscope and then assay the drop for Rous sarcoma virus using the assay I just showed you. And as it says here, the findings show that virus is produced at a slow trickle by a high proportion of the cells as they're maintained in culture. The cells remain alive as measured by the ability to continue producing virus. These characteristics distinguish this from lysogeny. And so this was important in terms of the paradigms, the mental pictures that people brought to the phenomena they were observing. I mean, so there was a precedent for a virus which could change the cells and could produce progeny virus, and that was phage lambda. But in phage lambda, as I'm sure you know, lambda can transduce genes into bacteria, and the bacteria are perfectly happy and no virus is produced, and then lambda can switch from this lysogenic, practically silent state to the vegetative lytic state. They are mutually exclusive. Growth of the cells and production of progeny virus is mutually exclusive with lambda, and that was the picture that people brought to a transforming virus, namely Rous. And this experiment by Ruben showed that no, it's something completely different that's going on with Rous. And then maybe the most important, foundational thing that Ruben did was this. So this was a paper published in 1958 by Temin and Ruben. And in this, what they did was work out the conditions to get foci of transformed cells in tissue culture, in petri dishes containing chick embryo fibroblasts. And this graph shows you that the number of foci that you get is directly linearly proportional to the amount of virus that you inoculated into the petri dish. That tells you that each focus is initiated by a single virus particle. Jonathan wanted me to explain 2-hit curves, but I said I was going to compromise and explain 1-hit curves. So this is a 1-hit curve where each focus arises by infection with a single particle. And the fact that you can do this, is really important because it means you now have the ability to count infectious particles of Rous sarcoma virus, and that's really the foundation for a good part of the rest of my talk is the ability to do this, and I'll mention that again. Now, as I said, now this is just a little bit of historical context. So, I want to say a little more about this. So this was done at Caltech. Temin was a grad student and Ruben was a postdoc under Delbecco's auspice at Caltech. And Caltech, of course, was one of the great centers of phage biology, the other being Cold Spring Harbor. But Caltech under Delbruck really created the modern biology. And the tool that they used to study phage was the plaque assay of phage. And Delbecco was a veteran of the phage group. He had done important phage research. And then he made a big move. He moved from bacterial viruses and bacteria to animal viruses. And he developed the plaque assay for poliovirus, which was tremendously important at the time. I mean, you can't start doing anything quantitative or analytical until you have an assay, until you can measure something. So Delbecco introduced the plaque assay, first plaque assay for an animal virus, which was polio. And this is really an extension of that. So in the plaque assay, whether it's phage or polio, you are counting little localized areas of cells that have been killed by localized spread from a single lytic virus. And what you're measuring in this assay is foci of transformed cells transformed by the transforming activity of a transforming virus. But it's the same idea. Now, as I said, I was here in 1961. In fact, don't tell the authorities, but I crashed the symposium in 1961. I came to Cold Spring Harbor early and crashed the symposium. And that may have been the most important, exciting moment in the history of our field. So here are some things that were discussed at the meeting for the first time. So Jacob and Minot presented and published in 1961 their synthesis of the behavior, genetic behavior of mutants controlling expression of beta-galactosidase in bacteria, and the genetics controlling lysogeny by phage lambda. And they realized these seemingly disparate phenomena could all be explained by the same basic model, which is you have a gene that makes something we call a repressor. The repressor goes and sits down somewhere else on the genome of the organism, which we call an operator. And next to the operator is the promoter, which controls expression of genes downstream from the promoter. And so the repressor essentially was the first transcription factor. And so this was really groundbreaking at the time. Also, messenger RNA, I think this was published in 1962 by Brenner, Jacob, and Messelson. So people had been suspecting for quite a while that, I mean, we knew that the genes were DNA, we knew proteins were made on ribosomes, ribosomes were full of RNA. Well, obviously, DNA gets copied into RNA, and that's the RNA in the ribosomes. And it was very important at the time to realize, no, the RNA, the bulk RNA in the ribosomes is not the informational RNA. There is this small amount of very short-lived in bacteria, different RNA that's actually the informational RNA. So that was messenger RNA. The genetic code was just beginning to be cracked by Nierenberg and Mattei. So Delbruck passed through Cold Spring Harbor while I was there, and Mattei came up from NIH and whispered a couple of letters to Delbruck, but it was classified, of course. Also, there was a lot of talk at Cold Spring, at the symposium, about what was called the Novick-Szilard-Umbarger effect, which we now call allostery, and was really described most elegantly by Minot, Wyman, and Changeux. But I digress. Back to Rous sarcoma. I told you Temin and Rubin had devised the focus assay, and I'm now going to tell you some stories, and I want to emphasize all the data for all the stories I'm about to tell you was obtained with the focus assay. That's all the data there was. Folks, there were no plasmids, no kits, no protein gels, no sequences, no clones, no westerns, northerns, southerns, PCR, transfection, mutants, just the number of foci that you got. And as it happened, I mean, this Rouse is a chicken virus, and there were also no permanent lines of chicken cells. So all the experiments were done on secondary cultures of chick embryo fibroblasts. So this is what that means. This was the routine in the lab. Every Monday and every Thursday, embryos, eggs containing, embryonated eggs would come into the lab, and the embryos would be taken out, minced up, trypsinized, and put into culture, and pooled, and the pool would be put into culture. Those were the primary cultures, and then, which was these pools made every Monday and Thursday, and every Thursday and Monday, these primary cultures would be retrypsinized and seeded into petri dishes. Those are the secondary cultures seeded into petri dishes to be used in experiments. Okay, and remember that they were pooled. So usually the routine worked fine, but some weeks something was wrong somewhere. You got no foci anywhere. It was eventually found, I mean, the mystery came to light eventually. It was found that some embryos contained a virus, produced a virus that made cells resistant to Rouse. The virus was called RIF, for resistance-inducing factor. Rubin published a paper about it. And then Rubin, together with Peter Vogt, a postdoc in his lab, I mean, we're continuing trying to work out something about the virology of Rouse sarcoma virus. And one thing it seemed important to do was essentially biologically clone the virus. That is, obtain a culture of cells infected with only a single particle of Rouse sarcoma virus instead of a heterogeneous mixture of Rouse transformed cells. So they infected cultures near the dilution endpoint, and they found, oops, in addition to Rouse, there was another virus in the Rouse stocks, and the virus did not induce foci, but was detectable because, like RIF, it rendered cells resistant to transformation by Rouse. They named it Rouse-associated virus, published a paper about it. This is Rubin and Peter Vogt, probably taken here in the early 60s. This is a relatively much more recent picture of Peter, who is still running a very active, productive lab at Scripps. And so they followed this up. I mean, so now they had found that they didn't have a pure stock of Rouse sarcoma virus. They had a mixture of two viruses. Rouse was contaminated with this other virus. What was it doing there? And what would a culture be like that was infected only with Rouse? So they set about, the next thing it was necessary to do was to isolate cells infected only with Rouse, not with both viruses. And this was done by Saburo Hanafusa and Teruko Hanafusa, Saburo's wife. They were postdocs in Rubin's lab. So they infected cells at very low multiplicity and cloned the cells. They picked clones of transformed cells and grew them up. And they got a big surprise. They found that these transformed cells were not making any infectious virus. These cells were called non-producer cells, and when they infected these cells with RAV, the cells released Rouse. In other words, Rouse was a replication defective transforming virus. RAV was always present in Rouse stocks because Rouse can't replicate without a RAV-like virus. RAV was a helper virus present in the stocks. They published this absolutely elegant paper, which I really recommend to anybody who has the time to read a beautiful piece of ancient history. This is just one bit of data from that paper. This is virus in the medium from a non-producer culture, and there is 0 virus in the medium until they added RAV to the culture, and then you get 5 to 6 logs in a day. This is Hanafusa, who is one of the heroes of the story I am telling you today, as is Peter Vogt, as is Reuben. And then, so that was very surprising. I mean, no one had expected that Rouse was a defective virus and that there were 2 viruses in the Rouse stock, since again, the only tool you have is foci of transformed cells. But then the plot thickened some more. Occasionally, you would get a Rouse stock, where a Rouse stock means a ground-up chicken tumor. Occasionally, you would get a Rouse stock which was able to overcome the resistance by RAV or RIF, was able to induce foci on cells infected with RIF or RAV, although these cells were resistant to most Rouse stocks. These unusual tumors were called, in the lab at least, aggressive rather than submissive. And Hanafusa figured out what was going on. Aggressive tumors contained a second RAV, quite distinct from the original RAV. The new RAV was called RAV2. So now you could have Rouse rescued with RAV1, or alternatively, you could have Rouse rescued with RAV2. Having the 2 different RAVs made it possible to sort out which properties of a Rouse stock were reflected the RAV that had rescued it. And it turned out that this included host range, neutralization properties, and cross interference. And I'm going to belabor this point for a minute, and you'll see why eventually. And maybe this is so obvious that it doesn't need to be belabored, but I don't know, not necessarily. So here's the experiment I'm going to tell you about in agonizing detail. So you start with a non-producer cell. You split it and infect one aliquot with RAV1 and the other aliquot with RAV2. Now you have 2 stocks of infectious Rouse, one rescued by RAV1, one rescued by RAV2. Now you take these 2 stocks and again infect cells at low multiplicity and isolate new non-producer cells. And then you can take these new non-producer cells and infect each new non-producer culture with either RAV1 or RAV2, and then determine the phenotype of Rouse rescued from these cells. And so this is a long story with a very simple point. So this is a table from this paper, and this is a table describing the results of the experiment I just told you. You start with Rouse rescued by RAV1, you make non-producers, you rescue virus from the non-producers with either RAV1 or RAV2. And it turns out if the virus was rescued with RAV1, then it cannot make foci on cells that are pre-infected with RAV1. That's the resistance by which RAV was originally detected. But it can make foci just fine on cells pre-infected with RAV2. Conversely, if you make your virus from the non-producers by super-infecting them with RAV2, it can make foci on cells pre-infected with RAV1, but not on cells pre-infected with RAV2. Now, similarly, if you started with Rouse made with RAV2, again, isolate non-producers, again, rescue from the non-producers with either RAV1 or RAV2, you get exactly the same picture. These 2 columns are the same as these 2 columns. In other words, the virus in this generation of virus has no memory. There is no imprint of which virus was started with only 1 generation ago. The phenotype, in other words, of the Rouse is completely temporary, transitory. It simply reflects the identity of the RAV that rescued it in this last generation. So what can we call this? How can we describe this, phenotype of the virus, but it doesn't belong to the virus? It's a coat that it put on today, different from the coat it might have put on last week. So we were standing around in the lab, a bunch of us, and talking about, so how can we, what's a good word? How can we describe this? And my Bratt, my old friend, Mike Bratt, he said, well, how about pseudotype? And that is where the word pseudotype came from, ladies and gentlemen. And I want you to know Mike Bratt is a very old friend of mine, and if you don't believe me, I have documentary evidence. That's us. Okay, so what? And now the history is going to speed up. Now we're going to do it more decade by decade than year by year. That is, I'm just going to mention some milestones in the history of the field. One of them, of course, is the discovery of reverse transcriptase. And I would say that before 1970, when these two papers came out, before 1970, there was just a whole bunch of confusion, controversy, mystery, hand-waving, what was going on. And after 1970, kind of everything fell into, I mean, not everything, but there was a great deal of clarity introduced overnight by this one discovery. I mean, then it was clear that, yes, there is RNA in the particle, and when it infects a cell, that RNA is copied into DNA. That followed from the discovery of reverse transcriptase. Just a little bit of context. I mean, how did these two people make this discovery independently, simultaneously? I mean, I don't know how come it was so simultaneous, but David Baltimore had been, in the 1960s, it was discovered that viruses could contain enzymes. Before that, a virus was just a protein package containing some nucleic acid. But it was quite important to realize, no, viruses can contain enzymes as well. And David was working out, you know, which viruses would have which kinds of enzymes and what was the logic determining what kind of enzyme would be in what kind of virus. And so it's pretty clear why it occurred to him to look for a DNA polymerase in preparations of what we called RNA tumor viruses. Meanwhile, Howard Temin had been maintaining for probably nearly a decade, against all odds, that although, yes, there's RNA in the particle, but there is a DNA form in the cell. And so really the two of them simultaneously finally did such a simple experiment. You know, you take a virus preparation, lyse it gently, and look for an activity that will incorporate deoxynucleoside triphosphates into a high molecular weight product. That was it. And so looking back, you know, from my many decades in the field, I feel that you can divide the history of the field into before reverse transcriptase and after reverse transcriptase. Another milestone that emerged from studies of Rous sarcoma virus was the discovery, identification, isolation of oncogenes. And this was done in part by Peter Duisberg and Peter Vogt and others. So it turned out that Rous sarcoma virus, the transforming virus, and RAV, it became possible to characterize the RNAs of the viruses in a little more detail. Peter did some beautiful work working out those techniques. And then he showed that Rous sarcoma virus RAV had a genome very similar to that of Rous sarcoma virus, except Rous had some extra sequences at the 3' end, not shared with RAV. And as Peter put it, A equals B plus X, and X was the Sarc gene. And, you know, many people had been interested in what were called RNA tumor viruses because of the transforming ability, obviously. I mean, the indication that there is something, some gene in this virus that somehow turns a cell into a cancer cell. And so those were the viral oncogenes, of which the first to be discovered was Sark. Many people who had been studying the viruses in search of how they transform cells, I mean, once you had the oncogene, you didn't really need the virus anymore. And so many people left the field of virology, took their oncogenes and went and studied signal transduction or whatever they do. And, and attendance at this field was declining. Of course, another thing that was happening over these couple of decades was that the tools that I told you were not available in the 60s gradually became available, and so we gradually learned more and more about structure and replication mechanisms of what we're beginning to call retroviruses, especially, of course, recombinant DNA technology, which came to the field around 1980 at the end of the 70s, and I would say revolutionized our field just like everything else in biology. Just a bit of hindsight about the story I told you a few minutes ago. So we now understand that what Rav donated to Raus was the ONF protein. In other words, just a little more background. So as it happened, the isolate of Raus sarcomavirus that was used in Rubin's lab was called Brian high titer strain of Raus sarcomavirus. And it turns out, we now know, this has a GAG gene, a POL gene, but its ENV gene is incomplete. And then, of course, it had the Sark gene. And so the reason it needed a helper was to complement the defect in envelope specifically. And envelope determines receptor usage, that is, in cells, this is, I think, generally true, in cells pre-infected with a retrovirus, the receptor for that virus is saturated by the envelope protein being made by the virus inside the cell. So that was the basis for the interference by which RIF and RAV were detected. And ironically, as it turns out, some Raus isolates are not defective, like Schmidt-Ruppin Raus is not defective. It has good GAG, POL, ONV, and Sark. And those are the outliers. Those are the exceptions. All other acute transforming viruses, retroviruses, are replication defective. That is, the addition of a transforming gene has been accompanied by the loss of some replication function. Okay, I'm now going to just mention another milestone, a tragic milestone in the field, which was around 1980, the early 80s. Gay men started coming down with Kaposi sarcoma and a whole plethora of infections, which the rest of us just slough off without even noticing because we have competent immune systems. And what was happening, of course, was that these guys were coming down with AIDS and they no longer had a competent immune system. And I just must tell you, this was absolutely terrifying. You could look in the textbooks, there's nothing there. This has never been seen before, this syndrome. What the hell is going on? And you can see that here in Lancet. Just kind of grasping at straws. What is going on? Multivariate analysis indicated independent significant associations for amyl nitrite. It's a happy times drug, and sexual activity, but not some other drugs. Also, of course, it wasn't just gay men. It was gay men. It was hemophiliacs who had received blood products. It was people who had received transfusions. It was people who had come from Haiti. I mean, there was a whole series of risk factors. And this was the death rate due to AIDS in the period from 1987 to 1994. But in 1983, Françoise Barré and her colleagues in Paris isolated a virus from lymphoid cells of AIDS patients, and in fact, as it says here, from a patient at risk for AIDS. And this virus, as in these cultures, looked like a retrovirus. And in fact, particles labeled with tritiated uridine, that is, they contained RNA, they contained reverse transcriptase, which is how she originally detected it. The particles band at 1.16 grams per mil in sucrose, just like a retrovirus. In other words, it was a retrovirus. And I know I don't need to tell you this. This was HIV, the retrovirus which causes AIDS. And once it was recognized as a retrovirus, the path to therapy was relatively clear. I don't mean to say it was easy, but, you know, it's a retrovirus. Okay, retroviruses have this enzyme, reverse transcriptase. We need to isolate drugs which will inhibit this enzyme. Retroviruses have protease. We need to isolate inhibitors of protease. And we know something about how to screen for compounds that will interfere with it, enzyme activity. So, you know, the virus was originally detected in 1983, and by 1995 in this country, HAART had been introduced and the death rate was dropping like a stone. Whereas what you might have imagined in this period was that it was just going to keep going through the roof because no one knew what was going on until 1983. So perhaps this message is unnecessary for this audience, but it bears saying anyway. And if you don't feel you need to hear it, please go home and tell your friends and relations and your congressman about the value of basic research. So I've been telling you about research on some viruses that were not human pathogens. So therefore, this was driven by the need to understand these organisms, not by trying to cure a human disease. And no one could have imagined HIV. No one could have anticipated HIV. So just try and imagine what it would have been like when these guys started showing up with AIDS and all these other opportunistic infections. There's nothing in the textbooks if there was not the background of knowledge of what a retrovirus is. I think the death rate would have continued going up through the roof. Okay, that's the end of my history part of the talk. I'm now going to spend a few minutes talking about viruses in general and retroviruses specifically. And perhaps, I'm sure this is familiar to many of you, but perhaps not all of you. So, but if this is about replication strategies of viruses, and as it says at the bottom, if you can imagine it, a virus does it. So virus particles may contain single-stranded DNA, they may contain double-stranded DNA, they may contain double-stranded DNA that was made from an RNA intermediate, that's hepatitis B. They may contain RNA that will be copied into a DNA intermediate, that's the viruses we study. They may contain double-stranded RNA, they may contain plus-strand RNA, that is, the particle really just carries an mRNA molecule. And they may contain RNA which is complementary to message. So you have all these, tremendous diversity of replication strategies. What they all have in common is they do all need to make an mRNA to tell the cell what the cell is supposed to do for them. And all these information transfers in all directions follow Watson-Crick rules. Viruses vary in complexity by 3 orders of magnitude, as far as we know. And that is in terms of nucleotides from a couple of thousand to a couple of million. Retroviruses are towards the simple end of this 3-log span, although not the simplest. It's very interesting to note that all those strategies I showed you on the last slide are all contained within the simple end of the spectrum. The largest genome that is not double-stranded DNA is that of coronaviruses, around 30 kb of plus-strand RNA. And everything bigger in viruses or cells is double-stranded DNA. Of course, viruses have a limited amount of genetic information with which to make multiple kinds of proteins, and so they do all kinds of tricks to do it. And retroviruses do some but not all of those tricks. So retroviruses make polyproteins, as do flaviviruses and picornaviruses and many others. They use alternative splicing, as we heard about this afternoon. They use translational suppression, which in turn can be either in-frame suppression, read-through suppression, that is translation of a stop codon as an amino acid, which is what gamma retroviruses do, as do alpha viruses, or ribosomal frame shifting, which most retroviruses do, as do coronaviruses and several other families of viruses. Retroviruses can use leaky scanning, that is a weak initiator, and many ribosomes, some ribosomes start at that weak initiator, but many continue past it and start at the second initiator. So one example is murine leukemia viruses. Gamma retroviruses make something called glycogag, which you've heard a little about, and glycogag is initiated from a CUG codon, 88 codons upstream of the GAG AUG. And other viruses also use leaky scanning. Retroviruses use internal promoters, and as do alpha viruses, and I'm thinking specifically of the HBZ protein of HTLV, the delta retrovirus, which we heard about from Charles Bangham today. And so HBZ is expressed on the minus strand from a promoter in the 3' LTR of HTLV, and HBZ is, so you have two overlapping reading frames. That's not that unusual in retroviruses or other viruses, except in this case, the two overlapping reading frames are in opposite directions on opposite strands. There are other tricks that some viruses use, but as far as we know, retroviruses don't. One is simply to have segmented genomes, that is, different genes are on different pieces of nucleic acid. So this is used by flu, reoviruses, and many plant viruses have irises, as do flaviviruses. And actually, it has been claimed that retroviruses do contain irises, and I would say this is not resolved. Another thing that orthomyxoviruses do is called termination reinitiation, where a UAA stop codon overlaps an AUG start codon. It's interesting, I think, to note that many of these strategies are used only by one or two, not all retroviral genera. In other words, even within the retrovirus family, different genera use different tricks, often to accomplish the same end. Okay, the place of viruses in the world of living things. So just relax. Viruses are the most abundant living things. Most viruses are in the oceans. If you took all the viruses in the oceans and lined them up end to end, they would stretch 66 times across the Milky Way. The majority of genes, that is, the majority of sequences in the biosphere, are in viruses. I don't expect you to believe me, but this is a fact. There's an Avogadro's number of infections every second in the oceans. And the turnover of metabolizing cells in the oceans due to infection by these lytic viruses is of such magnitude that it affects the global carbon cycle. And finally, some of life's persistent questions. Is a virus alive? What do you think, folks? Yes? How many votes yes? How many vote no? Really? Well, this kid would have dismissed the question. What a dumb question. Sorry, this kid. It's just a semantic question. You tell me your definition of alive, and I'll tell you whether a virus fits that definition. This guy, somewhat older and wiser, would have said yes, because they evolve. But this guy, one's forehead expands with increasing wisdom, and so This guy would be more emphatic about it. Of course they're alive. So what if they have an inert phase in their life cycle? So does anthrax bacillus. So do lots of plants, lots of invertebrates. Another of life's persistent questions: which came first, cells or viruses? And again, this kid would have known the answer for sure. Cells came first because viruses can't exist without cells, right? But it's not at all obvious to this guy. Yes, viruses as we know them today couldn't exist without cells. Cells are the niche in which contemporary viruses function, replicate. But maybe in the primordial soup, things like viruses emerged before or simultaneously with things like cells. So I don't know the answer to this persistent question, and I don't plan to take you through this forbidding diagram, but if you're interested, it's a review. It's from a review by Eugene Koonin, and he considers the question very seriously. The review is called A Virocentric Perspective on the Evolution of Life. Okay, finally, giving credit where credit is due. I want to just acknowledge my college genetics teacher, Tahir Risky. I still love Drosophila. I learned a great deal from Harry Rubin, including the importance of knowing the difference between what you think and what you know, and who introduced me to retroviruses. Sheldon Penman, I'm sorry I don't have a picture of him, but he was my postdoctoral mentor. And I must say, many of my principal memories of my postdoctoral tenure have to do with the war in Vietnam, but Sheldon had been a particle physicist who went into biology, and I think he went into biology very successfully, and I feel I learned from him the idea. It sounds simple, but I did learn it from him, that when you walk into your biology lab, you don't have to forget all the physics you ever knew. So I took a bit of a detour from virology, and I came back in the mid-'70s with the help of Bob Basson. And Bob was really the most generous, supportive mentor anyone could hope for. This is a very recent picture of Bob taken in Cuba. He just came back from Cuba a couple of weeks ago. Here's another picture from his time in Cuba. And Bob and I did a lot of biology on FV1 restriction in the mid-'70s. Here's the paper with the two hit curves that Jonathan wanted me to explain, but maybe one of us can do it at the bar. At the time, FV1 was a singular phenomenon, as no other restriction factors had yet been described. I'm also very grateful to Ray Gilden and George Vandervood, my first bosses in the early days at Frederick, who let me do my research and left me alone to do it. I'm sorry I don't have a picture of Ray Gilden. And I was extremely fortunate in Frederick in landing essentially across the hall or associated with Lou Henderson and Steve Roslin, because Lou Henderson and Steve Roslin and Alan Schultz were studying the experts at the proteins of retroviruses. They were real terrific protein chemists, and so they discovered myristylation of retroviral gag, which was found first in murine leukemia virus, and of course, a couple of years later on HIV. They noticed that there was this absolutely conserved spacing of cysteines and histidines in retroviral nucleocapsid proteins. They found that murine leukemia virus transmembrane protein is cleaved during virus maturation. The last 16 residues at the C-terminus of murine leukemia virus transmembrane are cleaved off by the viral protease along with the cleavages of GAG and POL. And by sequencing the N-terminus of murine leukemia virus protease, they showed that POL in murine leukemia virus is expressed by, as I mentioned a few minutes ago, translation of the stop codon at the end of GAG as an amino acid and continuing into the POL frame. Again, all done on the proteins, not by Sanger sequencing and staring at the sequences. And in each of these 4 cases, these discoveries were an opportunity to make interesting, informative mutants. And so very briefly, together with Alan Schultz, I made a mutation of the N-terminal glycine of murine leukemia virus GAG gene. And yes, the protein then was not burstylated, and that protein was not membrane associated, and there was no virus produced. So these conserved spacings of cysteines and histidines in nucleocapsid proteins were reminiscent of zinc fingers, which had been described in transcription factors. And so with Rob Gorlick, a postdoc, and Lou, we made point mutations in those conserved cysteines and histidines, both in murine leukemia virus and then a couple of years later in HIV. And it turned out the mutants where the cysteines or histidines had been replaced assembled okay, but they didn't package their genome. And so we did this first with murine leukemia virus. Then Rob wanted to do it with HIV. And I said, what do you want to do it with HIV for? I mean, you do the same experiment on different virus, you get the same answer. But Rob wanted to do it anyway, and so he did it. And I was right. He did the same experiment on HIV, he got the same answer, and he was right, because he got a job. We also made a mutant. So I told you that the last 16 residues of transmembrane protein of MLV are removed during virus maturation, the so-called R peptide. So we made an informative mutant by putting a stop codon at that cleavage site so that this mutant virus only made the mature short form instead of first making the long form and then truncating it during virus maturation. And we saw these humongous syncytia, and that told us that this C-terminal tail on transmembrane protein seems to be a safety catch, suppressing fusogenicity of ONV until the virus is released and matures. I think this is probably, I guess, an adaptation of murine leukemia virus to keep its host cell happy. These viruses coexist with their host cell for indefinitely, and I think murine leukemia virus has no wish to damage its host cell. And finally, as I mentioned, I studied together with Judith Levin and Yashun Feng the translational suppression between GAG and POL in murine leukemia virus. So one thing we did was show that not only was UAG translated as glutamine, but that UAA and UGA could also be translated in the viral context, and we identified the amino acids inserted in response to UAA and UGA. And we principally, Yashun, analyzed the signal in the viral genome responsible for the suppression. This is Judith and Yashun. And finally, it's getting on 20 years now that I have been privileged to work in the HIV DRP, and I've had these 2 bosses, which made the place a wonderful place to work, a stimulating, exciting, refreshing place to work. You don't need a picture of them because they're always in the front row here. And now I'm getting a 3rd boss, Eric Fried, and I have every reason to look forward to more of the same from Eric. So thanks to all.
Okay, well, Alan's happy to have any questions if anybody's got anything they want to ask.
So what is RAD 0?
Rab0 is an endogenous avian leukosis virus.
Was this the same skirt you were wearing actually at one of the first pictures?
What? Same
Shirt.
shirt? I don't think so. But I don't remember.
I was going to ask you what your favorite discovery or what your favorite experiment was.
I don't know. I love all my children.
If there's nothing, anybody want anything else?
What?
Thank you for showing all those figures in the old paper 1970 or
Oh,
before
yeah.
RT Discovery. I was curious why there's no error bar in the figure.
No errors back then.
Well, what are we doing wrong now? Okay, well, I think we can continue talking to you later.
All right.